Before I start, I'd like to thank the organisers for inviting us along. I think this is the first Shares Magazine event that we've done. I'm Stephen Franklin, Chief Executive of Evgen Pharma, and we trade under the ticker EVG. One very long disclaimer, which has been summarised already, and then on to our company snapshot. So, Evgen Pharma. We are a clinical stage drug development company. So that means that we have a drug in the clinic in real patients being tested as I speak. Our focus is in cancer, oncology, and neurology. We have a very capital efficient business model. So we are a virtual drug development company. So we don't employ lots of people wearing lab coats, buying very expensive centrifuges. We've got a relatively small headcount and everything is outsourced. Our whole business has been built around this very interesting molecule called sulforaphane. And sulforaphane, as you will see shortly, is a molecule of botanical origin. So it was first discovered in plants. And we are developing this molecule as an anti-cancer and as a neuroprotectant agent. The big challenge with sulforaphane, as you'll see, whilst it comes from plants, it's incredibly unstable. So whilst it's shown great clinical promise, nobody has been able to make a product to be able to advance it into the clinic to be able to test it. Our technology is called Sulforadex, and this has enabled us to both synthesize and scale manufacture a stabilised sulforaphane. So for the first time, we can take this into clinical trials. Our lead product is called SFX01, and now that is in phase 2 for a rare type of stroke called subarachnoid hemorrhage. And it is this year later going to go into a phase 2 in the advanced form of breast cancer called metastatic breast cancer. And like all good pharma propositions, you have to have an absolute robust intellectual property position, because ultimately this is the thing that you're going to be selling on to big pharma, be that by a license deal or by an outright acquisition. So this gentleman on the left, who doesn't come through very clearly, he's much lighter than that in real life. This is Professor Paul Talalay from Johns Hopkins University, and this is the gentleman that discovered sulforaphane. So it was long known from the 1980s that people that ate brassica vegetables, such as broccoli, had a lower incidence of certain cancers. And that was established in the 1980s, but it was in 1992 when this gentleman worked out what the molecule was that was conferring the anti-cancer benefit. And it was a clever discovery because the molecule sulforaphane isn't even in broccoli. What is in broccoli is this very stable, larger molecule called glucoraphanin. And what happens when you eat the vegetable matter, the sulforaphane is produced at very low and variable levels down in the GI tract from your own microbes that live down there. So you're effectively making, at very low levels, an anti-cancer agent when you consume this classification of vegetables. And of course, this is the reason why kids hate these kind of vegetables. They've got high levels of these sulfur-containing compounds. So it doesn't matter whether it's cabbage or it's rocket or it's broccoli. It's got a very distinctive peppery type taste, and that's these sulfur compounds. So it was discovered in 1992, and then you can see the huge raft of scientific publications, initially as an anti-cancer agent, and then some 7 to 8 years ago, people were doing experiments in models of stroke, multiple sclerosis, Alzheimer's and Parkinson's, and its neuroprotective qualities came to the fore. So we had a molecule here with huge potential as an anti-cancer agent and a neuroprotectant, but nobody could still make this stable molecule. Just a quick chat about how it works. This is as sciencey as it gets. So this is sulforaphane. It binds to a protein called KEAP1. Now, KEAP1 drives 2 key fundamental gene pathways. It turns on the genes in green, and this is the major antioxidant response element. So in all chronic diseases, the cells there's an enormous amount of oxidative stress, and that underpins all diseases, whether it's COPD, osteoarthritis. By turning on these some 200 genes, you are enhancing the body's own ability to deal with chronic disease. On the right-hand side, it suppresses a whole panel of genes associated with cancer and inflammation. And that's called NF-kappa beta. So we are both an NRF2 activator and an NF-kappa beta inhibitor. And these are known to pharmaceutical companies, and there are many companies out there trying to find new NRF2 activators or indeed NF-kappa beta inhibitors. Our unique play in cancer on the right is that not only do we kill tumour cells, but we kill cancer stem cells. So there's lots of therapies out there, whether it's chemo or hormonal agents that kill tumours. That's not the problem. The problem is when you obliterate the tumour, there are cells left which become the tumours of tomorrow. They just regrow. So it's a bit like cutting down the tree and not getting rid of the roots. So the big advance in cancer therapy will be when you can treat the tumour and in parallel kill cancer stem cells. And that's what our agent does in cancer. So we have a promising molecule. Scientists have been pumping out what's called preclinical data since 1992. You can buy sulforaphane. It comes in a little glass, like that little glass vial, very expensive. It has to be held at -20 under inert gas. So you can imagine scientists can take this out of the freezer, they can do experiments, they can show that it works in various mouse models. So there's evidence out there for lung cancer, MS, Alzheimer's, but nobody can take it into man. Because clearly if you're going to go into the clinic, you need a stable pill-type format that you could reliably take. And also you want something with attractive costs of goods. You can't have a liquid that has to be stored at -20 under inert gas, which is ridiculously expensive. So this is the technology. SFX01 is our lead product. We have found a way of synthesizing sulforaphane cost effectively. That's the molecule that sits in the center of this ring. And what happens is when it's fully formed, it becomes captured within a ring of sugar molecules, creating a scaffold. And the molecule which is synthesized is never exposed to the outside world, otherwise it would immediately break down. And it's encapsulated within the cyclodextrin structure. This gives us SFX01. There's a three-dimensional model there showing the bioactive ingredient sat within the sugar scaffold, and that fundamentally changes the stability of this molecule from hours to years, and also from a liquid at -20 to a pill that you can leave in the bathroom cabinet. Excellent pharmacokinetics, so that means that when you take the pill, it comes out of the stabilizing agent. There's no point in stabilizing it if when you swallow it, it doesn't come out. So it's fully bioavailable to the body and behaves indeed like regular sulforaphane. And we spent years putting this through animals to show that it's safe, and then on into humans to show that it's safe in studies called phase 1 trials. And we used the proceeds from our IPO in October of last year to advance the drug into patients for the very first time. So this is our pipeline. The breast cancer trial will start later this year. The subarachnoid hemorrhage trial has started, so the drug is now in patients, and I'll describe what subarachnoid hemorrhage is shortly. In MS, we're doing some interesting studies in preclinical, where we're comparing SFX01 to tecfidera, the leading drug in MS at the moment that's doing some 4 billion in sales. And we have some new analogues, so you can't just have this one asset. So we have in-licensed some technology from the University of Seville, where we've taken the core backbone of sulforaphane and changed some of the functional groups. So we have some selenium molecules, some fluoride molecules, and you can create a whole new generation, a whole new library of compounds from the one compound that nature gave us first, sulforaphane. So we have a whole raft of future compounds. So this is subarachnoid hemorrhage. Subarachnoid hemorrhage is a type of stroke, 5% of strokes in the UK. Unlike a regular stroke, of course, a regular stroke is a bit like a heart attack in the brain, it's a clot. This is an enormous hemorrhage. So what happens is an aneurysm pops, and you get this huge blood pour coming out, and you can see it here, it's all gone black. And it kills 50% of people there and then. You'll get a thunderclap headache, and it's horrible. For those that survive, they have a real challenge ahead of them. If they survive the initial bleed, they will be talking, they will be moving, they will be fine. But what happens 5 days later, or between 5 and 13 days, is they will get a delayed cerebral ischemia. So all that toxic blood that's left around the brain there will start closing down the blood pumping to the brain, and you will start to get early brain injury. And it happens 5 to 13 days after the initial bleed. So our interest is getting a neuroprotectant agent into the brain before the delayed cerebral ischemia to protect the damage such that these people can keep their speech, their emotional status, which is often impaired, and their movement. In 20 years, there has been no clinical development in subarachnoid hemorrhage. The last drug developed was a calcium channel blocker called nimodipine at the University of Southampton, in fact, where we are developing this drug. It is eligible for something called orphan status. Now, orphan status, to those of you who may not have heard of this terminology, because it's a rare disease, we have the capacity to fast-track the drug and get it to these patients that need it. So it's completely feasible that after our phase 2 trial, we could have a drug that could be approved because of its orphan status. It's a double-blind, placebo-controlled trial. It's on 90 patients, and as I said, the first person was recruited on the 30th of April. Annual sales potential for a rare disease, still significant. Different game in breast cancer. As I said, in breast cancer, one of the key challenges is you can, with hormone therapy, suppress the tumour. So most people that have metastatic breast cancer go on to hormone therapy. The problem is after about 9 months, you'll become resistant to the hormone therapy, and then you're looking at chemotherapy, etc. So what we're interested in doing is delivering SFX-01 on top of the hormone therapy to make the hormone therapy last for potentially years. So what you do is you turn this fatal condition into something that's akin to a chronic disease. So we're not trying to displace hormone therapy, we're trying to ensure that you don't become resistant to it, which is inevitable in all metastatic breast cancer patients. This is a trial that will start later this year. It's a multi-centre trial across Europe. We have 3 centres in the UK and we have 6 centres in Europe, so there's going to be a minimum of 9 centres. And some of the nuances of the trial design we are going to release in our year-end announcement, which is on the 31st of May. We're interested in tecfidera. It has the same mechanism of action. So we've got a £4 billion drug that's in the market now doing that level of sales. It's a small molecule, not great patent protection. It's an oral drug for MS. We hit the same mechanism of action 100 times stronger, and we believe we've got a better safety profile. So we're going after tecfidera, and we're doing that not by marching straight into a clinical trial, but by putting it head-to-head with tecfidera into some world-leading animal models. And that's good for our news flow as well, because as you'll see, the clinical trials report out after a significant period of time, and that's where your big value inflection comes from. But things like this, where we can potentially show that SFX-01 is as efficacious as tecfidera in animal models, is great for our news flow. We have lots of other collaborations in areas which are non-core, and we do this because we don't want to withhold drug from people. We like to control the IP, but we're happy to share material under material transfer agreements. So we've had publications in osteoarthritis where we've shown that SFX-01 maintains bone density. As you can see here on the right, this is a mouse model for osteoarthritis. And also very recently, we've got a new project, and this will be, the details will be announced at the year end. But this is with a European university where we're looking at Parkinson's disease. And in fact, Parkinson's disease and its link to sulforaphane as a potential therapeutic goes back to 2011. But again, nobody had a drug that they could advance into the clinic. I've touched upon this. Nature gave us sulforaphane. We need a pipeline of products. We can't just have one asset. So we're taking sulforaphane and we're doing medicinal chemistry around it. We have 60 new compounds. One of those could be better than the original parent. And it makes the proposition future-proof. Because if you look at drugs like statins, the first statin comes along, and before you know it, 3 or 4 years later, somebody else has got a new statin. It's essentially the same molecule with one group changed slightly better, slightly safer, and this is what happens with drugs. So you cut that off at the pass by controlling all the intellectual property at the front end for every derivative you can conceive of from your parent molecule. And we did this by doing a license deal with the medicinal chemists at the University of Seville that have a very strong patent position around sulforaphane and a whole raft of new analogues. So, in summary, we believe Evgen Pharma can unlock the therapeutic potential of sulforaphane. Nature gave us this molecule. Unfortunately, nature didn't give us a stable molecule. It's a molecule that if man wants to make, it's typically held under gas at -20. We have found a way of making that molecule cheaply and concurrently stabilising it in an alpha-cyclodextrin scaffold, creating a free-flowing powder that could be made into a capsule and pill and release sulforaphane to patient groups. This has enabled us to be the first company to take a sulforaphane molecule into patients. So all these years on, having shown that it's safe in animals and that it's safe in humans, we're now going to real patients in phase 2 trials, and phase 2 is the big value inflection point. Phase 2 is where we show the big pharmaceutical companies that we have something that works in patients. And that's what they need. Big pharma companies have an innovation deficit. They need to in-license assets that have shown that they work in man. Because what big pharma is good at doing is the big phase 3 and the marketing thereafter. And what they need is assets at this stage of development. And we would argue, if you look at drug development as a business, it typically takes some 340 drugs coming at that end of the pipeline to get one successful one out at the back end. So any company that comes along with 2 or 3 drugs, the odds are deeply stacked against you. It has to be said. And we manage that by saying, we don't have a new molecule. This molecule has been known since 1992 to be an anti-cancer agent. Everybody else has shown that in animal models. We know that it's a neuroprotectant. Other people and academics around the world have shown it works in models of Alzheimer's, Parkinson's, MS. What we have done is stabilise it and make it druggable for patients. So we would argue we have an atypical risk profile. And I think that is it. 4 minutes left.
Oh, brilliant. Oh, thanks, Stephen. Any questions, anybody? Gentlemen, just in second row and then come to you in the front.
Just one very dumb layman's question.
Yeah.
How much broccoli would I have to eat to have the same impact? It's not as simple as that, clearly, but I had to ask.
You have to ask. The Daily Mail likes to call us the broccoli company. I dislike it because we don't have anything to do with broccoli. But theoretically, you're at 2.6 kilograms if you're an average converter. You might require 10, somebody else might require 1. I don't advise going over 120 grams. You'll be very ill.
Gentleman at the front. Always the same question.
Well, yes, very similar question.
Oh, right.
Oh, I've got a question. So I've got the mic. I'll take the lead. I was just wondering, in terms of funding, so you said you had the IPO late last year.
Yeah.
Does that fully fund your stage 2 trials, or does that cover additional projects?
No, it's a good point, and I will take us back to the pipeline. So we did a pre-IPO investment of 2 million, which is an oversubscribed pre-IPO, and then we did back-to-back IPO raising 7, giving us 9 million in total. And the most important thing is we had to raise enough to be able to A, complete those phase 2 trials, B, assume they'll be delayed, and then have enough runway to do some kind of deal. So you're fully capitalised to not have to come back to the market to get to your value inflection. That's absolutely important. That's not to say that if the share price is at the right place and the MS data is looking fantastic, and there's the opportunity to raise more money, we would look at it. But we can go to value inflection on the cash that we've got. And that makes us quite atypical, I think, as a biotech company that will keep coming back to you folk and asking you for money every year. We can get to D-Day on our cash.
You said that if you took the right amount of, if you went for the broccoli rather than the drug itself, it would be something like 2.5 kilograms of broccoli you'd have to eat. If you could isolate the prodrug in broccoli, what sort of weight would you be? What would be stopping somebody just isolating the active prodrug? Sorry,
Right.
the prodrug rather than actually give the 2.5 kilograms of broccoli?
Number one, you have to buy a broccoli seed, you grow it through the agronomy, and then you extract it. So something between 0.5 and 1% of the mass of that vegetable is that molecule. If you go through the process of growing, harvesting, and then extracting, which incidentally I have done as a project, you can get a powder, and it's pure glucoraphanin. And then, of course, you could swallow that powder and hope that you're a converter. The problem is, by weight, it's more expensive than gold at that point. That's fundamentally the problem. Extracting from botanical sources is not economically viable. You could make glucoraphanin, theoretically, but it's quite a large, complex molecule to make. It's not a simple molecule like sulforaphane. But the biggest issue is, even with glucoraphanin, you don't know what dose you're getting. You can't do clinical trials and hope or guess that you're getting a dose. You'd need to do enormous trials to account for the natural variability, which is colossal. anything between 2% conversion and 40% conversion. So lots of reasons.
That's in humans.
In humans.
I see. I have another question, and that was about the calcium channel blocker you were talking
Yeah,
about.
nimodipine.
How effective is that in that indication
Poor.
versus no treatment?
Yes. So depending on which clinician you talk to, it's standard of care in the UK, and there is a clinical benefit, but the benefit is 5 to 6% improvement. But it's a generic drug now, so it's cheap. So you wouldn't withhold a cheap drug even with a 5 to 6% improvement. But the point is, there has been no development in 20 years, and what you need is a step change to keep these people talking and moving. And nimodipine's falling well short.
So you're talking about sort of 50% fatalities versus 45%?
No, I'm assuming that the 50% that didn't make it to hospital don't get treated with either nimodipine or with SFX01. What we're trying to do with the nimodipine at the moment is keep the blood vessels open and keep the blood flowing to the brain. That's the premise of how it's trying to work. Our drug works on a different mechanism of action and will be put on top of nimodipine. So we're not trying to displace nimodipine. That's quite important. You can't take patients off the current standard of care for ethical reasons. You have to leave it there.
So it's an add-on, effectively.
In the clinical trial, whether it ultimately displaces it because people are happy with, say, hypothetically, we have 60% improvement in patients and nimodipine could become displaced in due course. But for now, it's going to be an adjunct.
Last question, if I may. What was the potential market value for that indication? Was it? It was on the slide very, very briefly.
It depends who you believe. So that self-confession here, that is one of the largest numbers out there in the market. So we have a US peer that's trying to deliver nimodipine, not through a pill, but by leaving the tube in the brain. So you have your surgery and you have a tube coming out your head and they're pumping it into the brain. Their product is now in phase 3 and they, Credit Suisse have done a report saying the market could be $1.7 billion US. Our calculation is actually much more modest, could be 200 to 300 million pounds per year. But for a drug that could be approved on the back of phase 2 data, when we IPO'd and raised 5 million, it's still enough.
Any other questions?
I was quite intrigued as to how you recruit patients for the subarachnoid hemorrhage, because it sounds like it only arises once somebody's had a stroke, and you already said that it's likely people only live 5 to 20 days after.
We negotiated with the MHRA, which is the regulatory authority, something called assumed consent. So we're allowed to dose for 2 days, so they can get 2 doses. And thereafter, when they're conscious, you have to then have consent by either the patient themselves or the family thereafter. But we're very fortunate. It's such a serious disease with such a high fatality rate, and the drug is so safe because it's from broccoli that they're not going to necessarily withhold that from patients that could benefit. But it's a huge point. You've got people flown in a helicopter unconscious, and you've got to get the drug in very, very quickly, otherwise you miss the window.
You mentioned you signed up your first patient on the 30th of April. I presume they'd gone through the first treatment, have they, or first 2 treatments?
Yep, so the first patient came in and met the criteria on the 30th of April. There have been more, but I've promised not to get hung up on the number nor keep sharing the number with the market. It'll come out systematically as and when. But the key thing with all trials in recruitment is keeping the recruitment rates going. So many trials, people say it takes that long, and then they'll report back and say we're behind on recruitment, and all of a sudden what is a short trial becomes a long trial. So it's critically important that you keep on top of the recruitment, and the way you do that is you have other additional sites ready to turn on if you need them. So we have other sites ready to join Southampton. Southampton serves the whole of the south of England. Everybody's flown into Southampton, but if you get another one like Birmingham or Glasgow, you can really turn on that recruitment.
And can you adjust the dosage? And if you find under the first few patients that the dosage is too much or too little, can you adjust the dosage within the trial?
No, the dose is fixed, and we're delivering the top dose that we can deliver within our safety envelope. Right. So we showed in trials with healthy people that we can go up to 600 milligrams per day before you start getting the first side effects. So that's our starting point. So we are delivering 300 mgs twice a day. We know that's safe. We can't go any higher.
Anybody else some questions? Gentlemen, just over there.
How many employees do you have, and what are your sort of overheads annually?
It depends how you define an employee. In full-time equivalents, we're only 6 people. We have other people, NEDs, etc. You could argue there are 10 people on the payroll. This company is virtually fully there. There's one more position that we're looking at, which is a relatively junior scientific position. So, with 6 or 7 people, you can get a feel for what the monthly burn is. It's not, compared to most biotech companies, that horrendous, and you can actually turn the tap quite low. So when you get to the end of 2018, you can make a small amount of cash actually last quite a long time if you need to before you can get the deal going, because we've got no massive fixed overheads. We've got one small office in Liverpool, which is shorter than the distance between me and you. So very low overheads. And that's a key part of the model.
Gentleman just down the front again. Anybody else after?
You said that the dose would be 2 times 300 milligrams.
Yep.
Is that the little molecule in the middle?
No.
It's the whole thing?
Yeah, 1/7 of the enlarged molecule is the bioactive molecule. Yeah. So when we compare to literature, because all the literature is on the little molecule itself, you have to account for the fact that the bulk of it, by weight, is in fact the stabilising agent.
I was going to say, if it was 300 milligrams of a little molecule, it would be
quite a lot.
It would be a horse pill by the time you'd taken into account the cyclodextrin ring.
Yeah. Although a regular ibuprofen and paracetamol, people take 400 mg tablets. We're at 300 twice a day.
Any more questions, anybody? Gentleman down here.
Can you give a bit more detail on the IP protection globally and also the competition in this space, if there's any competitors that are similar, or if not?
Okay, so
similar product?
IP protection for a pharma company is key, because ultimately that's your asset. There is nothing else. When you do your deal with Big Pharma, you live or die by your IP and the strength of it. So we have multiple tiers of IP protection. Number one is what's called composition of matter. So that's protection for the composition itself. So sulforaphane, or an analogue of sulforaphane, contained within a cyclodextrin scaffold, of which there are a few different types, is our first claim. So it's new composition. And that's granted in territories such as USA, Canada, Australia, pending in places like Europe, Japan, and those will come through. So all the major pharmaceutical economies. Behind your composition of matter, you have process patents. So we have 2 process patents, which are about 18 months behind in terms of the prosecution. And we've used those as a means of going into all the manufacturing economies. So you cover off all the use economies, and you go into India and China and all these other places. So that's the third raft, the second raft. The third raft really is if you want to start changing the core structure of sulforaphane. That's a whole new patent family which we have with the University of Seville. And finally, the last bit of patent protection you have, which is what Biogen have for tecfidera, and they only have this. So they don't have composition of matter, they don't have process, but they have a formulation patent. And that basically says you deliver 300 grams of drug, which contains 200 grams of sugar, 100 grams of active, and it's encapsulated in a gelatin pill and enteric coated. And that's it. That's your final level. It's very specific. I would argue if you only had that, you're in quite a vulnerable position. There are other monopoly positions. When you get orphan designation, because it's a rare drug, you get marketing exclusivity. For up to 10 years, depending on the territory. It's very important. It stops generic companies copying you instantly. So it's not all about the patents if you're able to exploit these rare disease areas.
Can I ask a question about the company and its listing? You floated in 2015, raised £5 million. Mm-hmm. What is your market cap currently?
The current market cap, and I'm not one of these CEOs that looks every day. In fact, I look infrequently. The only market cap that I'm worried about is the one at the back end of that phase 2. And I encourage any investors to be the same. I wouldn't say this is a trading stock. You either believe the story and you believe the value inflection, or you don't. The idea of getting in and relying on some kind of small gain doesn't kind of really make sense to me. So the market cap, last time I looked, was 14 to 15 million pounds.
So its free float is quite good, basically.
Yeah.
So it's liquid stock. And farmers are driven by news flow. You've got a timetable here, obviously, for the rest of this year and next year. Looking over the next 3 months, 6 months, do you think there's going to be some news flow coming out?
Yeah, we have a news flow plan, and every 6 to 8 weeks, I'd like to think that we have a material bit of news. We're not going to be the kind of company that puts out puff and smoke and nonsense. Stephen Franklin got out of bed, RNS type announcements. It's got to be substantive. It'll be things like first patient in, a clinical trial approval. when you reach certain criteria, like the first 20 patients through subarachnoid trigger a safety review. I think that's a fair point to make an announcement, and then ultimately the results. But you also have regulatory announcements. So say you get orphan designation in the USA in subarachnoid, that's a big deal, you know. So there's a number of things, not least the MS data, which is going to come out this year as well. So there is a steady flow of news, but you can't get away from the fact that the big news that matters, that gives you the big multiple, is whether it works in one or both of the phase 2 trials. There's no escaping that.
A time for one more question. Anybody else? No? Stephen, thank you very much.
Thank
indeed.
you very much.
Thank you.